Hi student, I am Dr. D.S. Thaure. Now I share the screen of today's lecture in front of you. Please wait. Welcome to the 20th lecture of course number PAT 354, Diseases of Field and Horticultural Crops and their Management. So in today's lecture, we will discuss about Guava diseases. Okay, so wilt and anthracnose, this is the two most important diseases seen in the Guava. Okay, so wilt caused by the Fusarium of the Sporum forma species CD and anthracnose caused by the Glomerella CD. So see one by one, that is first one, fungal wilt. Okay, so already we see the different fruit crops suffering the wilt pathogen, that is a fusarium, and produces the external and internal type of the symptoms. Okay, for example, we already see the information about wilt of pomegranate and wilt of the also banana. So same symptoms, that is external and internal symptoms seen on the these seedlings of this guava and also the mature plants. Means initially drying of the leaf generally seen on the developing the seedlings as well as the mature guava plants. Okay, so this will be just a partial wilting type of the symptoms seen on only few wings and branches of the guava plant. So that's why this is known as the partial wilting symptoms. Then see these photographs. So in these photographs, these affected guava plants is totally wilted, and all the twigs and branches to be wilted, means whole plants to be dies. Okay. Then see these photographs. So some of the pinkish mass seen on the outer as well as inner surface of the vascular bundle cells. Means suppose this internal vascular bundles tissues seen. So that's why some brownish to blackish lines are seen. So this is this general external and internal symptoms also be seen in the guava plants caused by the Fusarium oxysporum or species CD. Okay, or okay. Then this Fusarium solani or Fusarium oxysporum forma species CD produces the two types of the conidia, macroconidia and microconidia. And this is the chlamydospores. So already I told with you regarding the morphological parts of the Fusarium oxysporum in respect to the Fusarium wilt of the pomegranate and Fusarium wilt of the banana. Okay. Then this is the perpetuation and this is the epidemiological parts regarding the wilt of the guava. And this is the management points. Okay, already we see this general information about the wilt diseases seen in the pomegranate and guava or maybe banana. So this is the same informations. Okay, then this is the second most important disease seen in the guava. These symptoms generally seen on the leaf, petioles, wings, branches, and fruit also. Means this pathogen attack to the all the aerial plant parts of the guava plants except root. Okay, so this is the most common or serious problem seen in the Uttar Pradesh, Punjab, and Karnataka, or nowadays this problem also be seen in the Maharashtra. So this anthracnose was first reported by the Mehta from Uttar Pradesh. So this anthracnose or these symptoms or this pathogen also be developed the dieback symptoms, wing-like symptoms, wither tip symptoms, or fruit spot symptoms. Okay, means initially the fungus attack to the leaf portion and wings portion or shoot portions. So on the leaf, the fungus causes necrotic lesions at the tip or margin of this leaf. Okay, so these lesions are usually gray in colors. Okay, so this fungus also be infect to the developing tender wings, and these tender wings also wither and dies from tip downward. Means this drying of the leaf or tip of the shoots from top portion to the downward direction. So that's why this symptom is known as the dieback symptoms. Okay, then most evident symptoms generally seen on the developing guava fruits. Okay, means pinhead-sized spots are firstly seen on the unripe fruits, which gradually enlarges over this surface of the fruits. Means these pinhead size dark brown to black lesions generally seen on these unripe fruits. So these spots become enlarges and to form the sunken circular black stomata. Okay, so you see these sunken spots. Okay, these sunken dark brown to black lesions generally develop on the unripe fruits and also the ripe fruits. Okay, so sometimes these whitish spore mass seen on these sunken lesions in the adverse or in the moist withers. Okay, so these several spots or this type of the several spots coalesce to form the bigger lesions. Okay, means the affected area in the unripe fruits become corky and harder. Okay, and in on the surface of the ripe fruits, the softening of the tissue and lesions generally having the 10 to 20 in size means this softening tissue generally spread to the outer surface of these ripe fruits. Okay, so these ripe fruits is unfit for consumption purpose because this tissue is already uh, infected and showing the softening tissues. Okay, means somewhat these dark brown spots become black in color and directly form the acervalized spore fruits. Okay, means acervalized spore fruits generally produced on these sunken blackish lesions may be produced on the unripe or maybe ripe fruits. Okay, so this is the symptoms developed uh, on the shoot portion, leaf portion, unripe fruits and ripe fruits. Okay, then causal organism that is Gliosporium CD or Glomerella CD. So this fungus develop the acervalized spore fruits. Acervalized means asexual spore fruits. So this is the asexual spore fruits in which develop the conaria and black city. Okay, means initially mycelium generally intercellular branch and light brown in colors. So this mycelium develop and to produce the conidiophore. So this conidiophore generally hyaline and small. Okay, so in between the conidiophores, the conidia and black city generally produced. Okay, means at the tip of the conidia core, the sickle septa unicellular uh, conidia generally produce. Okay, then perpetuation. So this fungus maybe perpetuates in the infected leaf, infected shoot, wings, and fruits. Means perpetuates in the dead parts of the wings, fruits, etc. Okay, this is the primary source. And secondary source, already conidia generally produced in the sunken spots or sunken lesions. In these conidia spread through wind or air currents. And last point, that is the management points. Okay, means in management points, collect the infected fruits seen on the guava plant. So collect such fruits and burn properly. Or collect the infected twigs or cut the infected twigs from or below the infection point. So in this way, you can avoid the primary source of infection or spread of the disease from one plant to the another plant. Okay, then spray the guava trees with the dipollutant 0.3% and dithin M78%, that is a 0.2%. So these two fungicides is also effective in controlling the diseases. Okay, so these anthracnose symptoms generally develop on the unripe fruits as well as the ripe fruits. So to protect the ripe fruits, kept in the storage conditions or in the transport conditions. So such harvested guava fruits immediately dipped in the uh, tetracycline solutions or maybe solution of the carbon enzymes. Okay, this to first prepare the 0.5% carbon enzyme solution or 100 ppm tetracycline solutions. Then the harvested guava fruits dipped in such a prepared solution for 20 minutes. So in this way, you can avoid the post-harvest losses caused due to the this fungus. Okay. Then also we use the borax mixture 1% or copper chloride 0.2% before the onset of the monsoon. So in this way, you can reduce the disease incidence. Okay, so this is the information about the anthracnose disease seen in the Java. So anthracnose symptoms generally seen in the field as well as the in storage conditions. This is the post-harvest disease also. Okay, so in today's lecture we discuss about the wilt and anthracnose disease seen in the Java. Okay, now I end the today's lecture. Thank you.